Welcome, everyone. Thank you so much for joining us today. I'm Tracy Smith, the Program Director of Education and Outreach for the Center for Integrated Cellular Analysis, based in New York City. In this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. So today, we will discuss new work using spatial RNA and protein profiling to understand how the tumor microenvironment drives macrophage heterogeneity and function in breast cancer. We'll have time at the end of the talk for questions, and we encourage you to either raise your hand, and I'll ask you to unmute yourself, or you can type your question into the chat, and I'll read it aloud to our speaker. We are really excited to have as our guest today, Nir Ben-Chetrit, a postdoctoral fellow in the Landau Lab at Weill Cornell. Nir earned his PhD at the Weizmann Institute of Science, studying the cell intrinsic mechanisms that propel breast cancer metastasis in patients with triple-negative tumors. He continued his studies of tumor immunology and progression at Memorial Sloan Kettering Cancer Center and Weill Cornell, and is interested in developing novel frameworks to untangle complex interactions and immune evasion mechanisms in the tumor microenvironment. With that, welcome, Nir. I'll hand it over to you.
Thanks, Tracy, for the kind introduction. So my name is Tracy mentioned, my name is Nir Ben-Chetrit. I did a postdoc in Dan Landau's lab at the Genome Center at the Cornell Medical College. And today we're presenting strategies that basically face a major obstacle to immunotherapies, primarily by the accumulation of tumor-associated macrophage in the tumor microenvironment and the effective immune suppression those macrophages have on the adaptive immunity. So I would start basically with the frame of the work. Targeted therapies are tailored to interfere with specific components of oncogenic pathways and achieve initial response in cancer patients. However, these strategies are limited by tumor evolution that is fueled by the combination of genetic heterogeneity and effective treatment, which eventually leads to therapeutic resistance and disease recurrence. However, immunotherapies offer an avenue to overcome this limitation by harnessing the immune system of the patients and by providing three main differentiating factors. First, they target the specificity, they target the protein product, not the activity of the other mutation. Second is memory. Once it's successfully recognized by the immune system, the protection is for life. And most importantly is adaptability. So once those approaches are successfully recognized in the tumor antigens, they're agnostic to tumor evolution, and the astronomical combination of BCR and TCR possibilities enable targeting of continuously evolving tumors. And indeed, the ability to restore effective immune surveillance in cancer patients has created a paradigm shift in immuno-oncology, particularly in patients with solid tumor. Mechanistically, those therapies are targeting restraining mechanism in T cells, unleashing remarkably effective and durable responses in refractory cancers. However, this unparalleled success was very limited to a very small subset of human cancer. And although the trial on the right eventually led to the approval of PD-1 and triple-negative breast cancer, those patients do not display the long-term remission seen in melanoma. This trial also provides very insightful data points. Even if you select for patients which have more target to more basically more target to tackle, we do not see an improvement of the efficacy even we increase the cutoff of the patient. Namely, we cannot really unlock the full potential of immunotherapies, and we maximize the effect of PD-1 in those cancers. So this is basically framed my research motivation and asking two major questions in tumor immunology. First, why do immunotherapies fail in most human cancer, and how can we overcome the failure mechanisms? So to get better understanding, we need to take a closer look at the cancer immunity cycles, which provide informative clues. This cycle is basically the fundamental process that generates tumor-reactive T cells. Now, since the checkpoint inhibitors repotentiate the adaptive arm of the immune system, there are two major dependencies that limit their efficacy. First, they require abundant new antigens, and those antigens are important to induce the T cell reactive T cells against the tumor. However, neoantigens are abundant in a very small subset of human cancer with high mutation burden, And since somatic mutation is the main source of tumor antigens, the lack of abundant mutation render most tumors, including breast cancer, polyimmunogenic. The second dependency is even if we have activation of T cells, once those T cells hit the tumor microenvironment, they rely on robust immune responses in the tumors. And basically, this is the major limitation because once the T cells are entering the tumor cells, the abundance of tumor-associated macrophage is basically rendering the tumor microenvironment completely immune silenced. And this activity of tumor-associated macrophages is basically highlight those cells as a major target for immunotherapy. However, most strategies today target one out of many immune modulating arms of the tumor-associated macrophage in tumor microenvironment. So Alberto Montevani first discovered the pro-tumorigenic activity of macrophages and actually coined the term tumor-associated macrophages, called them corrupt policemen, referring to the fact they're actually white blood cells that helping the tumor instead of killing them. And he also referred to the fact that macrophages can toggle between two activation states, namely the M1, in which they will support T cell and NK activation, and M2, which basically reflect the Th2 response, and will do exactly the opposite. They will silence the immune system and start repairing the tissue in a wound healing setting. Not surprisingly, the M2 state is hijacking the tumor, and the question is like how this state is being harnessed or hijacked by the tumor. And basically, it's capturing a very basic activation of macrophages that relates to wound healing repair. In a wound healing, in normal tissue, what you have usually, macrophages will respond very immediately to the wound and turn M1, clearing all the potential bacteria or whatever inside the tissue. However, once you remove those pathogens, they will enter repair mode. And in that repair mode, they will actually build back the tissue, activating fibroblasts, endothelial cells, and growth factors to stimulate epithelial growth. And once the M2 repair mode is finished, the site is basically resolved and everything goes to steady state. However, in the tumor setting, it's very different because the tumor is a wound that never heals. Macrophages are locked in this M2 or the repair mode, stimulating a variety of cellular processes, namely from growth factors, EMT, metastasis, immune suppression, and remodeling of the tissue, and fuel the tumor microenvironment and tumor progression. This limitation highlights the potential highlight the therapeutic potential to harness macrophage plasticity for innate immunotherapy. However, there is a very strong challenge. The mammary tumor hijacked the wound healing phenotype in a very complex and poorly understood mechanism. And what we are really missing is a scalable and spatial for TME modeling for high-throughput target discovery of tumor-associated macrophages. And for that purposes, we develop a method or what we call a platform signaling that we call organotypic TME. In that case, we are harvesting the tumor from animal. In that case, it's breast cancer. We remove the CD45 negative cells, CD45 positive cells, stabilize whatever is not immune cells, and then we restore back the immune component we would like to model. And this basically system recapitulates the composition we see in breast cancer, namely the flavors of the tumor cells, both luminal and basal, and the two flavors of fibroblasts, namely the ECM remodeling and the inflammatory fibroblasts that we see in the tumor microenvironment. This system also captures the structure, so we see the tumor nest, the stromal band, this build on top of each other. And most importantly, we can recapitulate the signal complexity that we see in the tumor microenvironment, as you can see in the cytokine, that each compartment from this organotypic system provide different cytokines. And altogether, we have the ECM component, which is critical for macrophage education. So first, we basically try to understand how macrophages are educated in that system. And once we add either monocytes or BMDMs from the bone marrow, we actually saw very nice recapitulation of the phenotype in terms of spatial localization. That's exactly what we see in the human tumor. We see the tumor nest macrophage and tumor-associated macrophages. And then by RNA-seq, we just basically verify that the transcriptional alteration in tumor macrophages, they adopt the wound healing response and they become proliferative. And this is more a functional study. So we try to understand or to confirm that these macrophages are become refractory to ATH1 signals, and that it's indeed what is the case. Once they educate in the system, they become refractory to LPS interferon gamma, so they cannot promote the TH1 response. Most importantly, we try to identify the phenotypic fidelity or to verify the phenotypic fidelity of those macrophages. Here we took single-cell RNA-seq from the organotypic TME macrophages, crossed-it on the x-axis with the tumor-associated macrophage from that specific tumor model in vivo. And then we project those signature into the human triple-negative breast cancer. And what we saw is a very nice recapitulation of the three major component tumor-associated micro subset that were identified in the study. And if we now try to project on how those signature correspond to survival, we can see that the macrophages signature that we generate ex vivo can recapitulate the poor survival feature that we see in human breast cancer, and this is using the TCGA. And this is after we normalize to CSF1R, namely that we are capturing the phenotype, but not the abundance of the macrophage, which are also critical prognostic marker for poor survival. So using that system, now we can interrogate like few parameters that we cannot do in vivo. For example, we can now study education dynamics, the complex crosstalk between tumor stroma and the immune system, and then most importantly, to apply perturbation. It is something we did in the Genome Center together with Neville Sanjana. I will not present the third part because of lack of time, but this is all the modalities we can apply. So using that system, we basically ask two fundamental questions of tumor, of macrophage and tumor biology, namely how they accumulate in situ and how they acquire the proteomogenic phenotype. And let's start with the accumulation. So it is well known across cancer and across patients that you have, once you have more macrophage in the TME, you have poor prognosis and those patients do badly. Macrophages are critically rely on the cytokine that promotes signaling, promote proliferation through the CSF1R. So the CSF1 comes from the tumor microenvironment, and the CSF1R is expressed on the macrophages. So basically, if you try to understand like what is the source of the macrophage, there are two main sources. One is the embryonic state. So in this route, the macrophages are preceding the organs, and they coming from the yolk cell. And after birth, you have always replenishing of the macrophages in the organ through the monocytes, and specifically through the recruitment of CCR2 and CCL2 axis. A few years ago, there was a hallmark study that tried to understand what is the source and the origin of the tumor-associated macrophage, and they basically confirmed that macrophage can come from either tissue resident or the monocyte using the CCR2 DTR. So basically, once you apply the DTR, you deplete everything that comes from the CCR, which is mainly monocytes, and they actually confirmed that macrophage in tumor in mammary tumor can originate both from the tumors and macrophage and monocyte. However, there is a very interesting data point that was completely missed by the authors. And basically, when they deplete or they ablate monocyte recruitment, so they could very effectively reduce the macrophage abundance in healthy tissue, but not in mammary tumor. And this raises interesting question because it basically says suggesting that the monocyte, the long-term accumulation of macrophage in tumor microenvironment actually rely on macrophage proliferation or self-renewal and not recruitment. And this is basically against the dogma. And this also raises very fundamental question in how macrophage or CSF1 signaling acting on macrophages. And since the mammary gland macrophages are primarily quiescent and CSF1R is abundantly expressed in healthy gland in tumor, it was unclear to us from the get-go why only TAMs can proliferate in response to the same hematogenic signals, namely why macrophage in the mammary gland CSF1 signal will provide survival mechanism, while in mammary tumor, CSF1R will provide proliferation signals. So luckily, the system captures exactly that. So if you look at the top row, you see tumor-naive macrophage in response to CSF1, they will not become proliferative. However, once we add macrophage to the system, they now start response to CSF1 and start proliferate. So then we ask, what is the pattern of this proliferation? This is just stochastic litigation between the EGF, between the CSF1R and CSF? And the answer was not. If we do EDU, BRDU, double pulse chase, we can basically score for the history of proliferation by the EDU. And then just hour before the analysis, we score for BRDU, and then we actually can trace back to ask, of those, the cells actually proliferate right now, what was the history of proliferation that happened 3 days before? And what we saw is that the same subset that actually proliferating while we do the assay had a long history of proliferation. Namely, the macrophage and tumor microenvironment has the characteristics of self-renewal. So to better understand how those macrophages gain this phenotype, we basically did a single cell time course trying to establish the transcription signature that give rise to this phenotype. And we did a time course with naive macrophages after 2 days and 10 days. And then we put all the transcriptome, single cell transcriptome on pseudo time and trajectory. And what we saw is that instead of going straight to the M2 phenotype, the macrophage has like an intermediate phase of interferon. And during this interferon phase, there is a spike of Ki67 and proliferation of those macrophages. And then we ask, is this conjunction of interferon signals and the proliferation are just like coincide timely or just like actually functional connected? We blocked interferon during the education, and we actually managed to effectively block the proliferation of macrophage during the education. And we did it using TYK2, which is a pan interferon signaling blocking, or specifically via STING. And then we went back to the single cell transcriptome and re-annotate, not based on time, but also the cell cycle state. And we try to identify those genes underlying the self-renewal subset. And we identified I6A or SCAR1. And we verified in the system that SCAR1 actually labeling a very specific subset of F480 high SCAR1 positive macrophage and F480 low SCAR1 negative cells. And we tried to do the same profiling in vivo. We actually verified that the SCAR1 positive are the proliferating subset in mammary tumor. And then we asked, where are those, what are the cell-cell interactions that drive the SCAR1 F480 high macrophages? And what we did, we went back to the organotypic system, and we used macrophages that come from the tomato macrophages. So these are, the tomato will label the whole macrophage regardless of the F480 state. And then we stand with F480 trying to capture the F480 high and F480 low. We actually managed to capture these two signals. And when we add a fibroblast marker, we actually noticed that the F480 high exclusively sitting on the tumor stroma, not in the tumor nest. So we verified this by Ki67. We actually saw that indeed the stroma-associated macrophage, or the SAMs, are the proliferating subset, while the tumor nest macrophages are not proliferating. And we thought, okay, maybe this is because only the stroma is secreting CSF1 and the tumor nest tumor cells are not, but that was not the case. So it's the same. So basically, even the same, even if both fibroblast and tumor nest macrophages can actually sense MCSF, only the stroma associated macrophage can proliferate. And we verified this in multiple sources of macrophage, either from normal gland or mammary tumor. And we saw basically the same response. Only stroma-associated macrophages are proliferating, and the tumor nest macrophage cannot. So then we ask, what is the immune profile of those stroma-associated macrophage and tumor nest macrophage? And for that, we co-injected either pure epithelial cells to generate TAM and SAMs, or basically a tumor nest and tumor-associated macrophages, or tumor cells that were enriched with stroma cells to try to enrich for the stroma-associated macrophage fraction. And that's what we see in the bottom panel. Once you enrich for the fibroblast, you get like specific subset of stroma-associated macrophages, namely the F480 high that we capture in the system was consistent in vivo. We validate this also histologically. We can see that F480 high are those who actually proliferating. This is by Ki67 and also BRDU labeling. And also in human patient, we can see the same phenotype, only the stroma-associated macrophage are like the Ki67 in triple negative breast cancer. And consistent with self-renewal, we actually saw that macrophages start to proliferate even before it's a fully developed adenocarcinoma. And interesting, when you have a fully adenocarcinoma, those proliferating subsets actually synchronize when the tumor is proliferating. Namely, when you pick up areas that are Ki-67 high in the tumor, the macrophage will also be proliferative. Namely, the macrophage and the tumor cells proliferate together. And then we ask, okay, what is the mechanism? How does this signal is being licensed? So we thought the story was basically solved, but it was not. And when we actually tried to co-culture fibroblasts, naive fibroblasts from the mammary gland and macrophages, we did not see any response unless we activate the fibroblast by the addition of tumor-associated tumor epithelial cells. And even if we stimulate tumor-naive macrophage with interferon, we still do not get any proliferation. Namely, we have a complex interaction between the tumor stroma, the tumor cells, and the macrophage to license macrophage proliferation. And again, this was not dependent on MCSF signaling. And so basically what we try to do is to ask whether, sorry, I forgot to mention this. So we basically saw that IL-6 is induced once we add the three components, so the stroma, the tumor cell, and the macrophages. And then we use ADAM17 inhibitors that block IL-6. And we actually saw that once we block IL-6, we can really effectively reduce the macrophage proliferation in the tumor microenvironment. And because secreted factor was not enough to induce the macrophage proliferation, we checked if this is made by cell contact. And we actually saw that even if we separate the cells physically by transwell, we cannot produce the robust proliferation of macrophage. However, when we let cell contact, those macrophages start to proliferate. And just to make a long story short, we look for cell contact-mediated signalings. We identify Notch, specifically Notch4, as one of the regulators of this phenotype. And when we inhibit Notch4 in vivo, we actually reduce the macrophage proliferation in the tumor, but also block proliferation of the tumor. And when we checked in a different model, in that case, ovarian cancer, it's not only blocking, actually reducing, inducing a tumor regression. So basically, just to conclude that part, we identified that in the mammary gland, CSF1 is transducing survival signals unless the fibroblasts are not active. In the tumor setting, and those tumor nest macrophages are just in survival mode by the CSF. However, once you induce the activation of fibroblasts by the IL-6 in conjunction with interferon gamma and Notch4 will now induce self-renewal of the macrophage and accumulation of these cells in the tumor microenvironment. So then we ask, beyond the self-renewal, can those cell-cell interaction induce a different phenotype? So basically, just to highlight what we try to understand here is, given the M1 and 2 paradigm, can those interaction can flip the microenvironment, depends on the cell-cell localization? And the answer that is basically yes. So to try to generate immune profiling in SAMs and tumor nest macrophage, we differentiate monocyte other in the presence of tumor nest of tumor epithelial cells on tumor stroma. And we can see that beyond the phenotypic, beyond the morphological differences of tumor nest macrophage and tumor stroma macrophage, we actually recapitulate what we see in patients. This is on the bottom, on the top bottom panel. And when we try to profile the immune activation status of those macrophages, we can see a mirror image. So stroma-associated macrophages are the suppressive, namely by the CD206, which is a M2 marker. But then they would be negative for the CD11C. However, the tumor nest macrophages are becoming inflammatory. This is marked by the CD11C and negative 206. And so beyond activation, the immune activation status, the stroma-associated macrophage are also the phagocytic one. This is like the main function of the macrophage, and SCAR1, which is like the marker we discovered before, is consistent with this immune profiling. And then we ask, is this preserved in human? And the answer is yes, and not only in tumor setting, but also in normal settings. So on the top, you see healthy mammary gland stained with 206, CD206, and 162, which is a pan-macrophage marker in human. And you can see very nicely that only the stroma-associated macrophage are positive at 206, which is the immunosuppressive marker, but also in the tumor setting. And we can actually see the mirror image or the reciprocal image in the CD11C, CD163. So only tumor nest macrophage are now stimulatory and positive CD11C. However, the stroma-associated macrophage will be exactly the opposite. And not only immunosuppression, but also function. Here we test cell scavenging. So we challenge the cells, either tumor nest macrophage, stroma-associated macrophage with T cells that are labeled with GFP. And we saw that basically most of the engulfing is occurred in the stroma-associated macrophage, not in the tumor nest. And this was in conjunction with the immune suppression marked by PDL1 and the immune activation marked by the CD11C. And then we ask a simple question: given the plasticity of macrophage, can the tumor microenvironment override this phenotype? Is this our plastic? And basically, we try to challenge an established model in the field. And there was like a paper published by Ido Amit and Stefan Jung a few years ago, showing that if you deplete the tumor-resident macrophage and now allow monocyte to repopulate the organs, those monocytes cannot recapitulate exactly the same phenotype that the tumor-resident macrophage have. And since then, the field is kind of basically, I would say, not stuck, but like I would say like they determined that the macrophage ontogeny is determined by the macrophage phenotype is determined by ontogeny. However, what we saw is very different. So here we try to flip the microenvironment. So first we generate tumor nest macrophage and tumor stroma macrophage, and then we flip the tumor microenvironment. And what we saw that this phenotype is very reversible. So we can actually flip from the SAM phenotype in the tumor nest phenotype in terms of the dendritic-like and the phagocytes. And not only morphology, but also functional-wise, you can take SAMs or immunosuppressive SAM and you can term them immunostimulatory and vice versa. And when we did the same experiment in vivo, we actually saw it was consistent with our hypothesis. So if we deplete the tumor, this is like a repopulation experiment in vivo, like what they did in healthy tissue. So we deplete the macrophage by CSF1R and let the monocyte basically rebound the tumors. And what we saw is basically the either monocyte that come from the host, which are MTMG positive. So basically everything is tomato will be the host, and those monocytes we adoptively transfer externally just to label the location. Basically, what we saw is that both monocytes and the tumor-resident macrophage can recapitulate the same phenotype regardless of the origin. And however, we also detect the safety signal because once those macrophages are being depleted and then you allow monocyte to come back, you gain only the suppressive phenotype and accelerated growth of this tumor. So this is basically a safety signal we try to raise, given that CSF1R antibodies are on basically clinical trial in patients. And then we ask this, how can we capture the heterogeneity in tissue? So for that purposes, we harnessed a method that we developed a year ago that integrates spatial transcriptomics and proteomics using the CICA technology. And in a nutshell, basically what we do is allow the spatial barcodes to capture both the RNA of those tissues, but also the poly-A coming from the ADT or from the tag that we have on the CICA antibodies. And what we did, we basically applied these spots into tumor tissues using a panel of 32 antibodies covering the lineage, immune activation, immune suppression states. And using deconvolution, we basically saw very distinct regions of this tumor, highlighting specific enrichment for cell types. And interestingly, once we apply deconvolution and clustering approach, we can actually segment the tissue based on the tumor evolution steps, starting from peritumor, hyperplasia, and early and late stage of adenocarcinoma, basically recapitulating the mammary gland transformation steps. And this also can be reaffirmed by the transcription signature of those regions. And then for the purposes of our study, we focused on tumor nest enriched region versus stroma enriched regions. And we actually confirmed that only regions enriched with stroma will have immune phenotype or suppressive immune phenotype in the macrophage, and vice versa. If you look at the cluster 2, which is enriched with tumor cells, you actually get stimulatory phenotype of those macrophages, confirming what we saw in the ex vivo. And we try to correlate the immune infiltration of T cells and NK cells into that region. We saw exactly the mirror image. So once you have suppressive macrophages in the tumor stroma, you have very poor infiltration of lymphocytes. However, this situation is flipped once you have tumor nest, immunostimulatory macrophage, and then you have very nice infiltration of T cells and NK cells into these regions. And now we can apply the RNA modality and confirm that actually the stroma-associated regions are enriched with interferon, exactly what we predicted in the model, and with the M2 phenotype and the wound healing that we is like basically a characteristic of those macrophages. And then we basically try to utilize single-cell RNA-seq from that specific model and project those single-cell transcriptomes of SAMs and TNMs back into the tissue, and we saw very nice correlation between the gene expression that we found by single cell and the gene transcription that we found in the spatial. Finally, what we took is we took those transcription signature and tried to project those into human breast cancer macrophage, and we identify the equivalent SAMs, the equivalent tumor nest macrophage, and then we actually confirmed this by staining. So C1q is a marker for the stroma-associated macrophages, and you can see the C1q is specifically expressed on the fibroblast areas, but not in tumor nest area. And then what we try to do is to try to correlate those signature into survival, and what we saw is that only the stroma-associated macrophages signature can actually predict poor survival in patients, but not the tumor nest macrophage, consistent with the phenotype. So basically, just to summarize, we actually challenging two major dogmas in the field. First, what is the signals that actually generate the long-term accumulation of macrophage? We show that macrophage accumulation is not relying on monocyte recruitment, it's actually relying on self-renewal, and we actually provide the mechanism for that, and also ways to target in patients or in mouse models. And we basically show also the tumor niches shape the macrophage functionality, overriding the ontogeny. So currently, the field is saying that macrophage phenotype is specifically determined by ontogeny. We show it's differently. And we provide the signal of action or signaling mechanism that license macrophage proliferation in tumors, so type I interferon, self-renewal, in conjunction with physical control of macrophage via the Notch4. And we show that immunosuppression and the immune activation of macrophages is basically spatially derived by the cell-cell interactions, and only SAMs can actually have the poor prognosis in patients. So just to summarize and to acknowledge all the people that actually help in doing this work. So two partners in crime, Steve, who is a brilliant bioinformatician, and Jesus is a very talented technician. We together led this project for a few years. And special thanks to Dan, which basically provide all the support and the help and the insight to continue and develop the story, and all the people across the street that help providing either samples and suggestions and samples. And thank you for the listening.
Great, incredible talk. Thank you so much, Nir, for sharing this with us. We'll now move on to our Q&A. So if you'd like to ask a question, you can raise your hand or type your question into the chat box and I can read it out aloud. But one question that I had is, so this work has all been done in breast cancer tumors. Do you think that this model of macrophage heterogeneity could apply to other tumor microenvironments, or is
Yeah,
the
actually, we saw very similar phenotype, let's say in lung cancer. We have now studied that it's a kind of a follow-up on the breast. And this is in humans. So we studied both the primary and the metastatic site in the brain. And we can really one-to-one project the signature that we have in the breast into the lung, because it basically refers to the cell-cell interaction. So we have either the stroma, which is almost identical, and also the other interaction with tumor epithelial cells.
Mm.
And we saw very, very similar response and very similar signature, almost one-to-one.
So going forward clinically, you're kind of turning, well, you've shifted this paradigm of thought that that the macrophages are, it's not just ontogeny. How is that being received?
Listen, it's a heavy lifting because we are actually going against the field. So we got response from the reviewers, and they kind of accept, but we need to put some more effort. And we know we are right because we use so many different methods, either by lineage tracing to show, yes, monocytes can enter the tissue, but they are not moving the needle. This is not what moves the needle when you talk about like accumulation. And once we perturbate with the Notch4, we basically eliminate or reduce the number of macrophages in tumor microenvironment. So basically, referring to the point that actually they rely on proliferation, not recruitment. And yeah, so let's see. It's going against the dogma is always challenging, but I think we have a compelling evidence. So
well, wonderful. Thank you for sharing that, and thank you again for being here. It's been very exciting. The seminar has been recorded and will be available soon on YouTube and on our website, multimodalintegration.org. And you can also follow us on Twitter at segs_ICA, or check our website for the latest announcements regarding upcoming talks and events. With that, I'd like to thank everyone for joining us today. Thank you again, Nir, and we hope to see you at our next seminar. Have a lovely day.
thank you.